Hello, this is Yerin, and today I'll be presenting my research on electroencephalography representation learning with spatial temporal reconstruction for accurate ADHD detection. To begin with, what is ADHD? Attention ADHD is attention deficit hyperactivity disorder, is a developmental disorder that encompasses a wide range of symptoms, including inattention, hyperactivity, and impulsivity that interfere with the body's functionality. The US has the highest number of adolescent ADHD diagnosis rate of 8.1%, but this problem is not just limited to the United States. The global prevalence of ADHD is around 6 to 7.5%. Despite its universality, the exact causes are unknown, but environmental reasons play crucial roles in its development. One crucial environmental factor comes from the advent of the digital era. During the COVID pandemic, as schools adopted online learning methods, adolescents were heavily exposed to electronics and faced hours of screen time. One study highlighted a positive correlation between screen time exposure and the development of ADHD, implying that the surge of media exposure continuously impacts adolescents worldwide. Unfortunately, there are various problems that should be addressed in ADHD. To begin with, conventional ADHD diagnosis method lacks scientific rigor because it heavily relies on diagnostic and statistical manual number 5 in this column, which is just a general checklist for ADHD symptoms. Since these checklists do not indicate specific biomarkers, they are far from being objective and objective. Furthermore, ADHD detection lacks accessibility. The detection of ADHD involves complex steps, a combination of questionnaires, and a doctoral confirmation. Consecutive medical visits may be unavailable for people who live far from cities or just do not have access to healthcare. The third reason is that the increased screen time exposure in adolescents may pose a risk to the development of ADHD, and some may just not be aware of their conditions. Taken all together, this set of problems calls for an objective biomarker for ADHD screening. As seen in the title, I incorporate a diagnostic test called electroencephalography, which is abbreviated as EEG, as the ADHD screening tool. The EEG data that are generated by its electrodes indicate specific parts where the brain was activated or deactivated using these wavelengths. It is helpful to determine abnormal brain conditions, namely ADHD. Since this technique is generally inaccessible for many, I propose to incorporate machine learning to train its effects. My proposed method addresses the various defects in the current medical field that I just mentioned. It takes EEG signals as input and produces the prediction of one having ADHD. In brief, it converts EEG signals into colored maps as shown here for the AI to classify by using convolutional neural networks. Convolutional neural networks, it looks like this. It means a type of artificial neural network primarily used for analyzing visual data like images and videos. By inserting these colored maps into the pre-trained convolutional neural network, namely CNN, another neurodevelopmental disorder classifier network called FCNN is formed, demonstrating one's probability of having a neurodevelopmental disorder, namely mine ADHD. So I obtained data from a publicly available dataset called AI Hub in 2024, and it is composed of 500 samples, which has 250 samples with no ADHD, which is the control group, and the other half with samples with ADHD. The age group for the sample is even from 0 to 18 years as shown in this graph, and which reduces potential biases in the section. To ensure the robustness and applicability of the proposed method, the dataset was split into 5 subgroups for five-fold cross-validation. To begin with, the first step of a proposed method is called pre-processing. The EEG divides its specific locations into 5 different lobes by using its electrodes, namely frontal, temporal, central, parietal, and occipital lobes. The EEG captures specific time steps, like in specific seconds, to analyze brain activity. In that specific time step, circular topological maps that look like this are generated and fitted into squares, generating an interpolated topological maps. These maps demonstrate the brain's electrical activity and classify sections into colors. So red basically means that it has high electrical activity, and the blue region is the area that has low electrical activity. These maps are generated to be used as inputs to the AI to facilitate its detection. So the second step is called the EEG representation learning. So this system it's basically simple. The system removes one topological map at a specific time step and replaces with a null matrix as shown here. Just like how the PDF zip is like formed, you know how you click on it, it expands and it compresses again, like does the same thing again and again. And this is just how I use to predict the probability. So basically using a CNN called encoder, the method compresses all data from topological maps, including the null matrix. Then next step at the reconstruction time step, the CNN finds the removed layer of the map, followed by another CNN called decoder, and then converts the null matrix into a layer that would be expected at that specific time step. So basically, after these steps, the method creates reconstructed topological maps that resemble the actual EEG data, which is the essential step of EEG representation. You guys might ask here, how did you actually train the AI to ensure its accuracy? So I trained the AI using a weighted reconstruction map that looks like this. So which helps detect how this is like a loss function which helps detect how accurately it uses the standard deviation curve, which balances out errors by penalizing values that are too far off. This helps accurately monitor the preciseness of the proposed method. So the last step is the transfer learning step. After the second step where I trained the AI, I then created a neural developmental disorder CNN that ultimately predicts ADHD screening. Instead of training the ADHD screening network from scratch, this transfer learning approach enhances accuracy and speeds up conversions. Here I use a binary cross entropy loss function. The original entropy function only measures whether the prediction was similar to the actual data, but the binary cross entropy loss function that I use measures the difference between the actual and predicted results, then also penalizes the approach if the difference is large, which also is a step to ensure high accuracy. So by using 5 different subgroups for patients, and I obtained experimental data. I used 4 input metric values, namely accuracy, precision, recall, and F1, and I used 3 CNNs called exception, confident 50, and resonant 152. In this, I basically determined the effectiveness of the proposed method by comparing results obtained using EEG representation learning and those without its usage, and I compared how much the accuracy dropped. So without EEG representation learning, all 3 CNNs show a lack of performance compared to results with EEG learning, signifying that the usage of EEG representation learning is quintessential. I then generated a confusion matrix of the highest performing CNN called ResNet 152 in this column. So it had a highest accuracy of 97% accuracy, and this map helps evaluate the performance of this method, and its performance in ADHD patients was about 99.31% and 94.78% for the control group without ADHD. So both values that are above 94 imply that the model accurately detects ADHD. So the last experiment is I just did it for fun, is to see which part of the brain most likely correlates with ADHD detection. I determined by excluding specific lobe every trial and saw which lobe correlates with the highest accuracy drop. The graph indicates that the exclusion of the frontal lobe heavily impacts the accuracy of the model, suggesting that the frontal lobe has the highest correlation with ADHD screening. So in conclusion, I propose a novel approach to detect ADHD using data obtained from EEG and establishing spatial temporal reconstruction by utilizing machine learning using convolutional neural networks. Implementing this reconstruction of representation learning, the experiment results reveal that highest performing CNN, resonant 152, generates an average of 97.04 accuracy in detecting CNN. My method will contribute to the development of more reliable indicator of the ADHD. In addition to developing a model to detect ADHD, the last piece of research comes from the brain anatomy-driven experiment, where the results demonstrate that the frontal lobe has the highest correlation to ADHD. One limitation to the study comes from data collection where subjects were all Korean. As future work, further developments can take place. In order to increase its broad applicability in different school settings where adolescents need accurate biomarkers to detect ADHD, this technique can be used universally. So this adoption of technique ultimately indicates that my project will facilitate better ADHD detection and early intervention for students and the general public. Here are my references, and thank you for listening.